This article is from nature.com. I will link it in the description. Protein misfolding and degenerative diseases. An error in protein conformation can lead to disease. What are the genetic and molecular causes for incorrectly formed proteins? Current advances in medicine and technology are making our lives longer. Sadly, as our life expectancy increases, the chances of getting a degenerative disease like Alzheimer's, Parkinson's, or diabetes also increases. Why is this? As incredible as it might sound, these diseases are caused not by bacteria or viruses, but rather by something conceptually quite simple: incorrect protein folding. Introductory biology courses teach us that proteins are essential for the organism because they participate in virtually every process within the cell. Therefore, if their function is impaired, the consequences can be devastating. As we age, mutations in thermodynamics, as well as some external factors, conspire against us, resulting in the misfolding of proteins. How does this happen? What are the genetic and molecular causes for incorrect folding of proteins, and what is their relationship to aging? Protein folding in the three-dimensional structure. Our modern understanding of how proteins function comes from almost 200 years of biochemical studies. Biochemistry is the science that studies the chemical processes in living organisms. Using different experimental models, biochemists demonstrated that most of the cell's chemical reactions and structural components are mediated or supplied by proteins. These experiments revealed that proteins are crucial for proper cell function. Actually, the word protein comes from the Greek proteos, which means first or foremost, reflecting the importance of these molecules. In 1917, the German chemist Hermann Staudinger proposed that organic molecules such as proteins were organized in polymers, giant molecules made of small molecule constituents linked together by chemical bonds in long chains. This idea contradicted the prevailing hypothesis, and it took some years for biochemists to accept it. Today, researchers know that proteins are long polymers made out of a set of 20 small constituents called amino acids. How are proteins made in the cell? The answer to this question took decades of study and the birth of an entirely new scientific discipline: molecular biology. Many experiments had shown that DNA is the vehicle of genetic information, and that DNA contained the information needed to make proteins. While discovering that DNA itself is a long polymer made out of 4 different types of small molecules called nucleotides, scientists realized that genetic information is transferred from a language system of 4 letters, nucleotides, in DNA to a language system of 20 amino acids in proteins. The anogenic funnel. The structure of a gene is one-dimensional. This means that a linear sequence of nucleotides codes for a specific linear sequence of amino acids linked to each other in a head-to-tail amino-carboxyl manner. The process of converting the information contained in the nucleotides to amino acids using the genetic code is called translation. Conceptually, translation expands the concentrated single dimension of the genetic code into a fully realized 3-dimensional protein structure. From this point of view, DNA in the genome are very similar to a highly compressed digital file, such as an MP3, in which a lot of information is packed very efficiently. How is this possible, though? The Nobel laureate Christian B. Anderson postulated an answer. He proposed that all the information needed for a protein to fold into its 3-dimensional conformation is contained in the amino acid sequence. To test his hypothesis, Anderson applied extreme chemical conditions to unfold an enzyme. These extreme conditions were called denaturing and were created with substances like urea, which at high concentration disrupts the non-covalent bonds of proteins, and mersapoethanol, which reduces diphide bonds. What happens to a protein exposed to denaturing conditions? As the primary bonds that hold the protein's 3-dimensional structure are disrupted, the protein unfolds. Later, after restoring the natural cellular conditions, Anderson observed that the enzyme's amino acid structure refolded spontaneously into its original form. He concluded that the native, or natural, conformation of a protein occurs because this particular shape is thermodynamically the most stable in the intercellular environment. That is, like everything else in nature, proteins achieve the lowest energy state possible. In other words, from the physiochemical point of view of a protein, the amino acids pack in such a way that the free energy of the molecule arrives at a minimum. Amino acids have different side chains, or R groups, which give them different properties. Some of these side chains are big, some are small, and some are hydrophilic, interact with water, and some are hydrophobic, tend not to interact with water molecules. Some are positively charged, and some are negatively charged. In a properly folded protein, hydrophobic amino acid residues are together, shielding each other from water molecules. Hydrophilic residues are exposed on the surface of the protein, interacting with the water of the cytoplasm, and big amino acids make nooks and crannies for small ones. This kind of tight folding and packing minimizes the overall free energy of the protein. An average protein has 300 amino acid residues. If we consider that there are 20 different amino acids, the combined number of protein sequences that can be made is astronomically high. By the most conservative calculation, the human body synthesizes at least 30,000 different kinds of proteins. Furthermore, the number of possible minimal energy configurations of a single protein sequence is also unimaginably enormous, and usually only a few may have normal activity. Surprisingly, newly synthesized proteins usually fold correctly in the appropriate minimal energy configuration, and thus they are able to do their job correctly. As Anderson demonstrated, the information needed for proteins to fold in their correct minimal energy configuration is coded in the physiochemical properties of their amino acid sequence. Usually, a protein is capable of finding its functional or native state just by itself in a matter of microseconds. The concept of how proteins explore the enormous structural conformation space is known as Levinthal's paradox. In 1968, Levinthal proposed that a protein folds rapidly because its constituent amino acids interact locally, thus limiting the conformational space that the protein has to explore and forcing the protein to follow a funnel-like energy landscape that allows itself to fold into the most stable configuration possible. Most proteins follow the correct funnel, but some of them have bifurcating pathways that make them fold in very different but energetically minimal structures, and only one of those is the native conformation. In these cases, something must come to their aid, helping them find the corrective native form. Amazingly, the rescuer is nothing less than a protein itself. Chaperones. Proteins that have a particularly complicated or unstable conformation sometimes have difficulty achieving their native state. In these cases, other, specialized proteins, called molecular chaperones, help them find their native functional conformation. Molecular chaperones were first mentioned in 1978 by Ron Lansky, who found that nucleoplasmin, a protein found in the nucleus of a cell, is able to bind to histones. Histones are nuclear proteins whose major function is to interact with DNA to form structures known as nucleosomes. Lansky observed that nucleoplasmin acted like a chaperone, accompanying and supervising the activity of the histones and preventing inappropriate interactions. Later, John Ellis extended this term chaperone to describe proteins that help other proteins fold or assemble into protein complexes. Interestingly, the existence of chaperones implies that some proteins have inherently unstable conformations that can flip from a functional minimal energy state to a state that is non-functional or even toxic. Why is the final conformation of a protein so important, though? The three-dimensional structure of a protein is what allows it to do its work, to connect with reactive sites on other proteins and molecules within the cell. In other words, the multidimensional structure determines its function, and this concept is one of the most fundamental within biology. Stable and unstable proteins. When native folded proteins are synthesized in a healthy cell, usually everything is right and well. However, our genome also codes for proteins that, as mentioned before, are inherently unstable because they have the property of folding in an alternative minimal energy states. Only very few of these alternative structures are functional and useful to the cell. The overwhelming majority are useless or even toxic. The functional or native conformation of non-membrane-bound proteins is typically water-soluble. Chaperones will help unstable proteins fold correctly, although some proteins misfold anyway. Misfolded proteins, also known as toxic conformations, are typically insoluble, but they tend to form long linear or fibrillar aggregates known as amyloid deposits. But how can a protein change so radically by folding differently if the sequence of amino acids is the same? The answer is in the way the amino acids interact. Protein conformation and the concept of misfolding. For many proteins, the most prominent structural motif of the functional protein in its native conformation is known as the alpha helix, a right-handed spiral coil. When a protein becomes toxic, an extensive conformational change occurs, and it acquires a motif known as the beta sheet. Note that the beta sheet conformation also exists in many functional native proteins, such as the immunoglobulins, but the transition from alpha helix to beta sheet is characteristic of amyloid deposits. The abnormal conformational transition from alpha helix to beta sheet exposes hydrophobic amino acid residues and promotes protein aggregation. As discussed already, misfolded proteins result when a protein follows the wrong folding pathway or energy-minimizing funnel, and misfolding can happen spontaneously. Most of the time, only the native conformation is produced in the cell, but as millions and millions of copies of each protein are made during our lifetimes, sometimes a random event occurs and one of these molecules follows the wrong path, changing into a toxic configuration. This kind of conformational change is most likely to occur in proteins that have repetitive amino acid motifs, such as polyglutamine, such as the case in Huntington's disease. Remarkably, the toxic configuration is often able to interact with other native copies of the same protein and catalyze their transition into a toxic state. Because of this ability, they are known as infective conformations. The newly made toxic proteins repeat this cycle in a self-sustaining loop, amplifying their toxicity and thus leading to a catastrophic effect that eventually kills the cell or impairs its function. A prime example of proteins that catalyze their own conformational change into the toxic form is prion proteins, discussed below. Under normal circumstances, the cell has mechanics to prevent proteins from folding incorrectly, as well as to get rid of misfolded proteins. Proteins that have problems achieving their native configuration are helped by chaperones to fold properly, using energy from ATP. Chaperones can evolve the Chaperones can avoid the change to beta sheet structure and aggregation of these alter proteins. Thus, they seem fundamental to the prevention of protein misfolding. Despite chaperone actions, some proteins still misfold, but there is a remedy. The misfolded proteins can be detected by quality control mechanisms in the cell that tags them to be sent to the cytoplasm, where they will be degraded. Infectious proteins. The concept of an infectious protein or prion was proposed in the 1960s to explain scrapie infection. Researchers found that the infectious agent that transmits scrapie is resistant to ultraviolet radiation, which typically destroys nucleic acids, and they proposed that this agent was actually protein-based. The idea that proteins could be infectious themselves was highly controversial because it appeared to challenge the central dogma of molecular biology. Eventually, Stanley B. Prosner and his team purified the prion protein responsible for scrapie, and they were able to show that proteins can indeed be infectious. For this work, Prosner was awarded the Nobel Prize in Physiology or Medicine in 1997. Prions are also responsible for the transmissional spongiform encephalitis, or TSEs, that include infectious diseases such as scrapie in sheep, bovine spongiform encephalopathy, or mad cow disease, whose infective form can cause Creutzfeldt-Jakob disease in humans, and Kuru, the only epidemic human prion disease known. In the late 1950s, before the idea of prions was even proposed, an epidemic of the neurodegenerative disease called Kuru suggested that proteins could be infectious. Kuru was discovered among populations of the Fore tribe of the eastern highland of Papua New Guinea, and the disease was associated with their cannibalistic funeral practices. With experimental testing, researchers showed that Kuru could be infective in chimpanzees after cerebral inoculation with brain suspension from Kuru patients. Years later, after Kuru was recognized as a prion disease, the discovery that in some conditions prions could be infectious across species led to the naming of a similar neurodegenerative disease, Creutzfeldt-Jakob disease. This affliction could be caused by the ingestion of beef containing toxic protein particles. The conformation error in the toxic protein can also be caused by a mutation, thus making the disease familial. Prions are not an exclusive phenomenon of mammals. They also occur naturally in unicellular organisms such as yeast, which therefore have become good experimental models for studying these protein conformational changes. Misfolded proteins and neurodegenerative diseases. Accumulation of misfolded proteins can cause disease, and unfortunately some of these diseases, known as amyloid diseases, are very common. The most prevalent one is Alzheimer's disease, which affects about 10% of the adult population over 65 years old in North America. Parkinson's disease and Huntington's disease have similar amyloid origins. These diseases can be sporadic, occurring without any family history, or familial, inherited. Regardless of the type, the risk of getting any of these diseases increases dramatically with age. The mechanistic explanation for the correlation is that as we age, or as a result of mutations, the delicate balance of the synthesis, folding, and degradation of proteins is perturbed, resulting in the production and accumulation of misfolded proteins that form aggregates. Among the environmental factors known to increase the risk of suffering degenerative diseases is exposure to substances that affect the mitochondria, increasing the amount of oxidative damage to proteins. However, it is clear that no single environmental factor determines the onset of these disorders. In addition, there are genetic factors. For example, in one of the simplest forms of familial Parkinson's disease, mutations are associated with the dormant forms of the disease. This means that an individual with a single copy of a defective gene will develop the disease, yet 2 copies of the defective gene are required for recessive forms of the disease to develop. In the case of Alzheimer's disease, and for other less common neurodegenerative diseases, the genetics can be even more complicated, since different mutations of the same gene and combinations of these mutations may differently affect disease risk. Misfolding in non-neurological diseases. Protein aggregation diseases are not exclusive to the central nervous system. They also appear in peripheral tissues. In general, the genes and protein products involved in these kinds of diseases are called amyloidogenic. Such diseases include type 2 diabetes, inherited cataracts, some forms of atherosclerosis, hemodialysis-related disorders, and short-chain amyloidosis, among many others. All these diseases have in common is the expression of a protein outside its normal context, leading to an irreversible change into a sticky conformation rich in beta sheets that make the protein's molecules interact with each other. The general pattern that emerges in all of these diseases is an abnormal tendency of proteins to aggregate as a result of misfolding. The aggregation can be caused by chance, by protein hyperphosphorylation, a condition, hyperphosphorylation, a condition where multiple phosphate groups are added to the protein, by prion self-catalytic conformational conversion, or by mutations that make the protein unstable. Aggregation can also be caused by an unrelated or pathological increase in the intracellular concentration of some of these proteins. Such imbalances in protein concentration can be a consequence of mutations such as duplications of the amyloidogenic gene or in changes of the protein's amino acid sequence. Imbalances can also be caused by deficiencies in the proteasome, the cellular machinery involved in the degradation of aging proteins. Inhibition of autophagy, a process in which cells engulf themselves, also promotes amyloid aggregation. In addition, some evidence suggests that the severity of these diseases correlates with an increase in oxidative stress, mitochondrial dysfunction, alteration of cytoplasmic membrane permeability, and abnormal calcium concentration. Looking forward. At the moment, there is no treatment for any of the known amyloid diseases. However, there is hope. The increasing knowledge of the causes of amyloid accumulation is beginning to pay off with possible pharmacological treatments. Therapeutic inhibition of precursor protein synthesis is well within reach with the expanding use of RNA interface technologies. Drugs that induce chaperone expression are also being tested, as well as inhibitors that prevent protein hyperphosphorylation, the thing where the phosphates do the thing. And as the number of known amyloid beta sheet structures grows, scientists have more options to find common structures for the design of specific chemical inhibitors of aggregation. Finally, vaccines against the aggregates are being developed. Although we are at risk of accumulating misfolding proteins every day we age, and to function properly our cells must continually make proteins, understanding misfolding will ultimately help protect us from serious diseases. And then there's references and recommended reading. Now, I'm probably not going to edit this. There's a couple of tables and things that I could put on the screen, but I'm probably not going to. I'm sorry for stumbling over my words so much, but no one's going to listen to this, so who gives a shit? Bye-bye.